Hello everyone, today we'll talk about the experimental proof for semi-conservative nature of DNA replication. Right, so the scientists who conducted these experiments were Matthew Meselson and Franklin Stahl. So we call this experiment as Meselson and Stahl's experiments. So what they did, they took a bacteria called E. coli, which is also known as Escherichia coli. They grew this bacteria on a medium containing N15H4Cl ammonium chloride. This N15 is nothing but the heavy isotope of nitrogen. Heavy isotope of nitrogen. They grew E. coli in this medium for several generations. For many generations. So what happened when they grew the same in this medium, N15 got incorporated. This nitrogen, heavy isotope of nitrogen got incorporated into the newly synthesized DNA. This DNA, the heavy DNA, can be differentiated from normal DNA by centrifugation in cesium chloride density gradient. What do we mean by this term, centrifugation in cesium chloride? So let's suppose this is the test tube and we have the medium here of cesium chloride. If we put the DNA here and this is the gravitational force, right? So heavy DNA would be here and light DNA would be above this, right? So this is the centrifugation when we do the centrifugation in cesium chloride. So this is what happened. This is the heavy DNA and this is the normal DNA, right? So after this, what they did after this step, they transferred these cells, the E. coli cells, which had N15 in their DNA, and they transferred this E. coli into a medium containing this N14Cl, NH4Cl. This N14 is the light isotope of nitrogen. So before going into the details, let me tell you one thing. So 20 minutes is the one generation of E. coli. In this 20 minutes, it can replicate its DNA completely. It needs 20 minutes for the replication of DNA. And when they transferred the E. coli into NH4Cl in the light isotope of nitrogen, so they took the readings at various time intervals. So they took the readings or the samples at various time intervals. So let me show the same how they took the readings. So earlier we had this N15 DNA. When they grew the same in N14 medium, in this medium, after 20 minutes, what they observed, that one strand of the DNA, this is the heavy, purple one is the heavy DNA. So what they observed, that this is they observed the hybrid form of DNA, which is the intermediate form of DNA as well, right? So after this, what they observed after 20 minutes in the same medium. Let me show you one more thing, that these are the two strands and we have the purple ones as well. These get separated and Now we have these strands. Right? So again, these are the intermediate ones or the hybrid ones, hybrid DNA, H DNA. And these two are the light DNA. So let me show you how we will be able to see this in the test tube. So here we'll have the light DNA in the cesium chloride medium. This is the intermediate DNA or the hybrid DNA. And here we'll have the light DNA. So I'm so sorry for this. This is the heavy DNA, because in the gravity we'll have heavy DNA over here, right? So this is the heavy DNA, this is the intermediate one, and this is the light DNA. I hope you understand this topic. If you have any questions, you can leave a comment. Thank you so much.